Welcome to this follow-up on the microbiology classes. Here we will come with a concentrated assembly of the information you've come, run through the most important things, so you have a kind of a scaffold to put on all the rest of your knowledge you have had from the previous classes and from your read in the book. The course is called Microbiology. One of the most important organisms and class of organisms in microbiology are the bacteria. Here I have a drawing of a standard bacteria. Bacteria are prokaryotes. That means they have, literally speaking, evolved before the development of the nucleus. I'll get back to eukaryotes in just a sec. The bacteria have a cell wall. Get back to that also in a second. They have a cell membrane, and in there, inside the bacteria, are some DNA. There's usually assembled in nucleoids, and it's not an organelle, it's just an area where the density of DNA is very high. They have ribosomes. They're more or less all over the place. I've just drawn a few. They can have, they don't have to, but usually they have some extra chromosomal pieces of DNA called plasmids. What they have of plasmids really depends on the conditions within which the bacteria are growing in. So they can disappear, they can come, they can be different ones, depending on where the bacteria are living and what are the evolutionary constraints on that bacteria. They have these pili in plural or pilus in singularis, which are small pieces, protein which sticks out. Get back to what they can be used for in a second. And they can move. They can have one or more flagella, or flagellum in singularis, which helps the bacteria to move. Not all bacteria has flagella, but some have. And finally, it can have some kind of particles, we could call it. We are not going to call it organelles. It could be, if it's a sulfur-reducing or oxidizing bacteria, a chemolithotroph. It can be sulfur. It can be gas vesicles in cyanobacteria, for example. It can be iron. It can be a number of things, which can also be present and can be spotted in bacteria. But basically, this is how a bacteria looks. And could actually replace this with Archaea. They are prokaryotes too. The general design is the same. We do not know as much about storage vesicles or storage particles, I should say, in Archaea. But apart from that, it's pretty much the same. The ribosomes are a bit different. They're more similar to eukaryote ribosomes, but they're still all over the place. They can have plasmids, so flagella and pilus. So the design overall of Archaea and bacteria are the same. Here we have a description of the cell walls in bacteria. You have the gram-positive and gram-negative. When we talk about bacteria, the easiest way to distinguish between different groups of bacteria is the gram staining. Is it gram-negative? Is it gram-positive? That's one of the first things you would do if you want to identify bacteria, is to gram stain it. Then you can go on to looking in a microscope, you can do chemical analysis, you can use different growth media. That's fine. But the first and vital importance, if you want to identify bacteria, is to make a gram stain. The gram-positive have one membrane and one cell wall. They have a very thick cell wall, and it has crosslinks, and it's made of peptidoglycan. Saying peptidoglycan means it's made up, basically to translate it is, it's made up of small proteins, peptides. Glycan is sugars. So a mixture of peptide and sugar. This thick cell wall keeps dye you use when you make the gram stain inside, and it will not get out again. And that's why it's called gram-positive. Gram was a Danish scientist back in the 19th century. Gram-negative have two membranes, an inner membrane phospholipids and an outer membrane. And in between is a thin cell wall. This thin cell wall is also made of a mixture of protein and sugar, so it's peptidoglycan again. The space between cell walls and cell membranes are called periplasmic spaces. Another thing about the gram-negative is they have a lot of molecules sitting here. They are, a lot of them are called LPS, lipopolysaccharides, mixture of lipid, fat, polysaccharides, sugars, a number of sugars sitting. And they will have them almost as a lawn on the outside. So these are how we distinguish between different groups of bacteria. Just to make your life as a student complicated, you have to remember some bacteria don't stain. They might not have a cell wall. There are a few of them, especially intercellular bacteria and bacteria living inside us, pathogens or parasites you would call them, that do not have a cell wall. Archaea have a cell wall too. It can be made in several different shapes. Generally they have some kind of pseudomorine, which is a kind of peptide sugar again, but different from the peptidoglycan. And they can have this glycoprotein layer, which is called the S layer. But this can be designed in many different ways. And we also find a few Archaea which does not have a cell wall, just to make things a little complicated. But if you remember S layer and pseudomorine, you know when you hear those words, we're talking about Archaea. That's good. Next step is the cell membrane. The cell membrane is made up by phospholipids. And it's a phosphate group sitting on an ester, and this ester is then attached to fat. They can be, sometimes, occasionally, can be unsaturated, meaning you have a double bond. It's a bilayer because the tails, which are hydrophobic, are up against each other, and then you have the phosphate groups, which are hydrophilic. So small area here. And this side could be the inside, and this side could be the outside, which are aqueous environments, so you need a hydrophilic head. This makes it possible for the cell, no matter if it's a bacteria or eukaryote, we have exactly the same design to control the flow of things in and out of the cell. You need to have channel proteins and everything. The only things that really pass readily through the membrane are carbon dioxide, oxygen, and water. Almost everything else needs specific channels or transporters to get in and out of the cell, which means you can actually have a gradient in concentration between the inside and the outside, depending on your needs. It can be a proton gradient, which can be used to generate energy. It can be a gradient of ions, as we have in our cells, with a lot of potassium inside, a lot of sodium outside, etc. But this membrane, the primary function is to control the environment within the cell, no matter if it's a bacteria or a eukaryote. Next is a brief introduction to the membranes in Archaea. Here we have the cell membrane from Archaea. The overall design is the same. So we have a hydrophilic head with phosphorus in there, both on the inside and the outside. But instead of having a lipid, it's built out of isoprene units. And the linkage between the phosphorus part and the isoprene part is an ether instead of an ester, as it is in eukaryotes and bacteria. Another thing we see in Archaea is that it can be a bilayer, as we see here, but it can actually also be a monolayer. And we can have some part of it can actually be like aromatic, like this, and there are a little more variation. A lot of this has to do with that we find Archaea in very extreme environments, especially extreme hot environments. And that needs the membrane to be designed precisely, because the lipid membrane we have, you should think of butter. And if you heat up butter to 100 degrees, it's a liquid. And if you're Archaea living in 100 degrees hot water, you can't have a membrane made out of liquid. You need to stabilize it, and that could be one of the ways. For both Archaea, eukaryotes, prokaryotes like bacteria, on the membrane, as I said, it creates an environment you can control, but also they sit a lot of proteins on the surface, which have different tasks. Some of them are, as I said, are channels or transporters. Some of them are involved in communication, in sensing, etc., etc. So the membrane is also, how should I say, a platform for a lot of different proteins. And now the eukaryote cell. Here we have a standard eukaryote cell. A lot of things has been left out for simplicity, so don't worry. Important thing is that we talk about eukaryote cells, we say they are made up of compartments. It's been compartmentalized. And the different compartments, most of them, at least in the eukaryote, are enclosed by membranes. Like the nucleus, mitochondria, LC is lysosomes, back to what that is, and PC is peroxisomes. Get back to that in a second. We also have the rough endoplasmatic reticulum and the smooth endoplasmatic reticulum. We have mitochondria and we have the Golgi apparatus. Little about the different compartments. First of all, the nucleus, that's where the DNA is stored. It's usually organized in linear chromosomes. Most of the chromosomes in bacteria and Archaea are circular. And there's not chromosomes, but of course there's only one, which is big maybe, but that's it. We, for example, have 23 pairs of chromosomes. Yeast have 16. We have the rough endoplasmatic reticulum. On the rough sits ribosomes. Inside the rough endoplasmatic reticulum, we have synthesis of proteins which are going to sit in a membrane or to be exported. The export function in a cell is made by the Golgi apparatus, named after an Italian, Camillo Golgi, or whatever you pronounce that. And this is really the post office, makes proteins ready to get excreted from the cell. Both in the rough endoplasmatic reticulum and in the Golgi apparatus, we also see modifications. Parts of the protein might be clipped out and something attached. And you will often see with excreted proteins that we attach sugars to them. Ribosomes is where the protein synthesis happens. Inside the cell, the nucleus, we will have transcription, where we make an RNA copy of the DNA. That's exported through the nuclear pores, which are quite big, out into the cytoplasm. And here it will enter the ribosomes and get translated. That's a process called translation into proteins. It can happen in the cytoplasm and it can happen in the rough endoplasmatic reticulum. The smooth endoplasmatic reticulum is primarily involved in the synthesis of lipids, for example, for cell membranes. But we have here a membrane, the rough endoplasmatic reticulum, ribosomes, and then membrane. Golgi apparatus is made of membrane. And of course, the cell membrane is made of membrane. We also have a membrane around the mitochondria, and inside the mitochondria we have an additional membrane which is made by the mitochondria itself. Mitochondria are prokaryotes, bacteria, who was caught 1.5 billion years ago or something like that. So in there are the green pieces here are actually mitochondrial DNA. They have their own DNA. And the red spots are ribosomes, but it's bacterial ribosomes. So different from the ribosomes we see out here. And you have had a lot of biochemistry, you know all about what happens with the electron transport chain, oxidative phosphorylation, which happens in the different membranes and the intermembrane sprays in the mitochondria. Not talking more about that. Lysosomes are small vesicles, membrane enclosed again. In there we have a different pH, usually 4 or 5, and we have a lot of proteases. And they are basically the recycling stations of the cell. So they recycle used up proteins, damaged proteins, are chopped up to amino acids there, and the amino acids can be used in the protein synthesis. Peroxisomes are involved in the destruction of things like peroxidases and superoxides, which can be happening there. There can be other compartments in other types of cells, so this is just a generalized picture. But most important about the eukaryote cell is that different processes happens in different compartments, and the compartments are enclosed by a membrane, a phospholipid bilayer. We find viruses in both eukaryotes, I have them over here, and bacteriophages is viruses that attack bacteria. There's also actually something called Archaea phages, which are the same. I'm not going to talk more about phages in Archaea, don't worry. In general, no matter if it's a bacteriophage or a virus, you will classify them based on 3 important things. What is the nucleic acid? Is it DNA? Is it RNA? Is it single-stranded or is it double-stranded? RNA viruses, there's an extra version. Is it a retrovirus? And retroviruses are viruses that have their nucleic acid as RNA, and when they get inside the cell, they will make a DNA copy of that we're using an enzyme called reverse transcriptase. HIV is a retrovirus. And then finally, the last version, does it have an envelope or not? Envelope is when the virus takes part of the host membrane with it when it leaves the cell. All viruses, both phages and animal viruses, are intracellular parasites. They are not live. They can be infectious and non-infectious. But you can't talk about viruses being alive because there's no processes. You can't measure any biochemical processes within a virus particle. They are a piece of protein, and out there sticks other proteins. So it's a protein box, you could call it, and inside this box is some nucleic acid, RNA, DNA. It can have, without an envelope, poliovirus is an example. It can have an envelope. HIV has an envelope. And here we have a fantastic drawing of a coronavirus, which also has an envelope. And out through, on the outside of viruses and phages, sits something which makes it possible for this virus to attach to somewhere specific on the surface on the host. This is what is called the virus receptor. The virus receptor is a protein with a completely different function, of course, for the cell. It can be communication with other immune cells, like the CD4, which HIV used to gain entrance. It can be ACE2, which is an angiotensin converting enzyme number 2, which is involved actually regulating our blood pressure, which sits in the airways and the lungs of us, and the nose actually too. Really depends. Viruses and phages use specific part in the host cell to gain interest. And when they get entrance into the cell, they take over the cell. The whole transcription, translation process is taken over by the virus, and the cell will start producing virus particles, which will become new viruses, leave the cell, and can infect neighbor cells, neighbor bacteria. Process is pretty similar, no matter, in general terms at least, if it's a virus or it's a phage. One thing that's special about bacteriophages is they can sometimes, instead of having their own DNA, RNA, they can pick up some of the host's DNA, RNA. And then when they infect the next cell, well, they will not be able to make new viruses because some of the genetic material is not virus, but it's from the previous host. But they can make work in transforming that bacteria because it will inject a new gene or whatever that might give the bacteria it infects new possibilities, at least change its genetic makeup. And that's a process we call horizontal gene transfer. You don't see that much in animal viruses. This part is primarily about bacteria. You could also say Archaea, but we concentrate on Archaea. And it is how they obtain the carbon, to a certain degree nitrogen, and energy. We use a lot of different labels. If an organism gets its carbon from CO2, we call it autotroph. An autotroph means that it gains some energy and uses that energy to obtain carbon dioxide, and usually uses that to make some kind of organic compound. I've just written the formula for glucose here. These down here, phototrophy, chemotrophy or chemoorganotrophy and chemolithotrophy, are related to the energy part. Where does the organism get its energy? Phototrophs are, when we talk about bacteria, it's cyanobacteria. Can also be higher plants and many of the green algae, or all the green algae and some of the other algae too, I should be precise. They get their energy from sunlight, and they have this protein called rubisco, and they can then convert the photoenergy, you could call it, into chemical energy, because this process here consumes enormous amounts of energy. Nitrogen can be obtained in three ways: from the air, from organic compounds, or from inorganic compounds. When it's air, it's N2, and nitrogen fixation is usually done by cyanobacteria. Nitrogen fixation usually happens in cyanobacteria, rhizobia, that's bacteria in the root nodules of peanuts, peas, clovers, alfalfa, etc., of Frankia, which are also in kind of the root nodules, but for example in some trees. This process is extremely energy consuming. Cyanobacteria gets the energy from life, so they are phototroph, autotroph, and they are able to fix nitrogen. However, oxygen stops the enzyme that fixes nitrogen. This enzyme that fixes nitrogen from the air is called nitrogenase, and oxygen turns off the nitrogenase, destroys the nitrogenase. So what cyanobacteria is doing is they make a special cell type, which is a very thick cell wall, which are called heterocysts, where this process of fixing nitrogen happens. Because one of the side products, when you autotroph and you fix autotroph, phototroph, and fix CO2, is a byproduct is oxygen. So that's how cyanobacteria do. Rhizobia and Frankia, they are chemoorganotrophs, and they get the energy from the plant, and in return they hand over nitrogen in the plant to the plant in the form of ammonia or nitrate. Okay, chemoorganotrophs. They are organisms that get the energy from organic compounds, like E. coli, which will import glucose, lactose, whatever. Pseudomonas, who will work on complicated organic compounds and destroy them. So they get it from organic compounds. They get the energy, and by that they will also get carbon, and they will also get nitrogen, because organic compounds contain those two atoms. Chemolithotrophs get the energy from inorganic compounds. For example, reducing or oxidizing sulfur, phosphorus, whatever, iron, and that energy can then be used so that you autotroph, or you can be a mixture or whatever. Most of the bacteria we meet in the lab are chemoorganotrophs. Most of the bacteria we know of and work with are something that live within us or in our vicinity. So they will be chemoorganotrophs.
Microbial growth and control. We need to know how to control the microbial growth. What is microbial growth? We have, of course, it's an increase of bacterial cells, the number of cells, and we also have the generation time, where the generation time is the time where from one bacteria to be two, or microorganism to be two. So it's duplicated. And how can they then have this increase of cells? Okay, we have two ways. We can talk about the budding. So we have the budding, and we also have this binary fission. So budding, we have the simple way here is when you have a cell, then it's starting up making a bud, making it bigger, and then it's divided into two, and then we have two cells. The binary fission, that's where we have a cell where it's duplicate the DNA, and then it's making this prolonges, and then it's divided here. It makes a septum, and then we have simply two cells afterwards. So that's the two ways all you have to know. And how can we look at this? How can we actually get this curve up so we can see if the microorganisms is in the lag phase, exponential phase, stationary death phase, or how it looks like for the microorganism? You can use a spectrophotometer. If you're using a spectrophotometer with the absorbance of 600 nanometer, so we have the absorbance for 600 nanometer, then you can see how the growth are, and then you can put it into this curve, and then you'll get a curve. For the budding and binary fission, we have budding, that's typical yeast. So you have Saccharomyces cerevisiae, which is using budding, and with binary fission, that's typical all the others. So that's it. Yes? That's with growth. Then we are going to look at the temperature. We can control the growth of microorganisms with several things, and temperature is one of the biggest one, where you can say that we have the temperature. And what is temperature? If I ask you about the cardinal temperatures, then you have to know what that is. So again, we have a curve with the growth here, and then we have the temperature down here. Then we have a figure like this. And down here we have the minimum. Over here we have the maximum temperature. And up here we have the optimal temperature. Each microorganism have its own minimum, maximum and optimum temperature. So remember that. So we have cardinal temperature is minimum, maximum. Then we have the temperature classes. And the temperature classes is, now I'm going to show you a graph of several, but that's just to show the structure of it. The microorganism have its own cardinal temperatures. So we're starting up here, again with the growth, then we have the temperature, and if we have about 0 degrees, 10, 20, etc. up here. Then we have the psychophiles. The psychophiles is at the cardinal temperatures in the lowest range. So you have it about here. So we have the psychophiles. The next is the mesophiles. The mesophiles is typical here, where you have its mesophiles. And now I'm just taking these away because it shouldn't confuse you. Because the psychophiles is typical around 0 to 10 degrees. Mesophiles is about 20 to 40 degrees. Then we have the thermophiles, looking like this, thermophiles, and they have perhaps optimum about 40, 50 degrees Celsius. And then we have the extreme thermophiles, which is away here, extreme thermophiles. So each range have a range of around 30 to 40 degrees Celsius. So that's why I'm just taking this away, because it's depending on each microorganism. So this is just this, to simplify and also show that the psychophiles is in the low end and the extreme thermophiles is in the high end. And then they are also overlapping each area, typical. So that's for the temperature classes of microorganisms. If you have to mention some of them, mesophiles is where most of those bacteria that we know, or microorganisms that we know and are working with in the lab, those are typical mesophiles. So that's where we are incubating about 20, 30, 40 degrees or 37 degrees Celsius. So that's where, E. coli and etc. are. Thermophiles is where we typically have those with the optimum about 50 degrees. That could be some of the lactic acid bacteria, which like to be around this 45, 50 degrees Celsius. The extreme thermophiles is typical Archaea, which is very extreme. They like to live in extreme temperatures, some of them. That's the temperature classes. There are several ways of controlling the growth of bacteria or microorganisms. And one of them is also pH values. So pH is also a major condition for the microorganisms. Neutral pH values is about pH 6 to 8. And then they are where we call the microorganisms neutrophiles. If they like to be in acidic conditions, we call them acidophiles. And if they are in the alkali area and like to be there, we call them alkaliphiles. We can put this range up where you're having the pH up here to 14 and down here 0. And here we have the 7 as a neutral. And if we're saying we have 6 and 8 here, this area is the neutrophiles. We are low, we have the acidophiles, and up here we have the alkaliphiles. Neutrophiles is, if I have to mention some of the bacteria, E. coli is around this condition. Acidophiles, we have typical, you can say, lactic acid bacteria. They are starting, the milk is around 6 to 7 in pH value, but they produce lactic acid, and then the environment will be a lower pH value, and they still like to be there and grow still until down to pH 3. So they are acidophiles. Alkaliphiles is some Pseudomonas, some Bacillus, and some what they are called methanogens, which I think you are going to look at in project 2. So this is about the pH values. Osmolarity is where we are looking at the available water. You can call the water activity available in the conditions or the environment for the microorganism. And water activity, also mentioned as A and W. If we're having pure water, then pure water is the, you can say like this, water activity for pure water is 1.00. If you have conditions where the pH value is lowered or higher, if you are lowering the water activity, you can do it with pH, sugar, salt, dry the environment so it's more dry. This is where you are going down to, let's say 0.62, where that's very few microorganisms that's able to grow there. When we are talking about water activity, I mentioned salt, and salt is also where the microorganisms is, some like to be in salty conditions with high salt concentrations, and some don't like that at all. So if we're starting looking at the halo conditions, we can say that halo is salty conditions, and we can mention a graph here. So we have the growth again, and then we have the salt concentration. So you can say salt concentration in percent, and typical we are having, like this, If we have 0 here, we have 1% concentration of salt, and then we have 5, 10, and then 20. We have some microorganisms which don't like to be in salty conditions, and they are starting up here and like to be in the environment, and then they find out there's too much salt, and then it's going down extremely quickly. So they don't like to be salty condition over approximately 1. So that's where we say it's halo or non-halo actually. Non-halophiles. That means they don't like salt conditions. Then we have the halotolerant. They like to be in, or they accept to be in the condition where there's salt available. And we can say it's like this, perhaps a bit higher, but I should do like this. And this is where we call it halotolerant. What's important to see here is that they like to be in the low end of the salt, but they still can grow in some salty conditions. We have also some who likes to be in the salty conditions. And they actually have a curve like this instead of. So they are those we call halophiles. They like to be in the area where there's a high salt condition. And then we also have the extreme halophiles. Typical around 10. They don't like to be under 10% of salt. So it's going up like this and then out. We call them extreme halophiles. There's not that many who likes to be in this area. Non-halophiles, I can mention E. coli. Halotolerant, I can mention Staphylococcus aureus. Halophiles, I can mention Alivibrio fischeri. And then we have the extreme halophiles. And we have one, it's called Halobacterium salinarum. And if you look at this name, you actually can see that it likes to be in salty conditions. We have the halo, which were the salt over here. And salinarum is also salty conditions, or like salt, actually means salt. So this is, sometimes you can look at the name of the microorganism and find out where they like to be. So Halobacterium salinarum likes to be in salty conditions. Also, another thing is very extreme, or not extreme, but we have this oxygen tolerance of microorganisms. Oxygen, some microorganisms like to be in oxygen or in aerobic area, and some don't. And so if we're just starting with this oxygen, if they are in the area where there's a lot of oxygen, we call them aerobic. And then we have also the anaerobic conditions. Aerobic is where there's oxygen, the bacteria or microorganisms is happy. If there's oxygen available where there's anaerobic microorganisms, they are not happy. So they like to be where there's no oxygen, then they're happy. We have to look at this and know that aerobic is where it's oxygen available, and anaerobic is where the oxygen is not available. And now we have to look at the different conditions for microorganisms. We have to describe the oxygen or anaerobic and aerobic conditions and where the bacteria or microorganisms like to be. We can mention it in those 5 classes here. If we have obligate aerobic microorganisms, they like to be in the top of the environment. So they like to be up here. And just to mention, to make it easy for you to understand, if we have the oxygen available up here, then it will be lowered. The concentration will be lowered within going down in this water. So if we have obligate aerobics, they like to be in the extreme high end of the oxygen available. The same for obligate anaerobic. That is, they don't like to be in the area where there's oxygen available, so they like to be in the bottom. What about the aerobic and anaerobic then? Shouldn't it be like this? No, because aerobic like to be in certain concentration of oxygen, so it's a bit down from the bottom, from the top to the center. So they like to be, and then it's less here. And for anaerobic, it's the same, just from the bottom, like this. So we have the aerobic, obligate aerobic, then we have the anaerobic and obligate anaerobic. What is this facultative then? Yeah, we have some called facultative, and that's where they are not that extreme aerobic or anaerobics. They like to be in a more widely area. So we have the aerobic, it's the red ones here. Facultative, they can live down here like this. And the anaerobic, the blue one then, they like to be in this area. And as you can see, they are mixing more or less. And that's where in this center is more or less where you can talk about the facultatives instead of just aerobic and anaerobic areas. So this is where we have the oxygen, and this is also where we can control how microorganisms, the growth of that. When we are in the lab and looking at microorganisms, if we are finding a sample in the environment where there's air available, then of course we have to incubate with oxygen or just in the normal air. If we are finding, taking a sample down in the soil, let's just say half a meter down in the soil, then probably we need to have it in the anaerobic conditions. So we have to remove the oxygen. So that's where also when we are having a sample, we have to look at where does it come from and how should we incubate it to make sure that we actually can see something on our plates. Microbial agents. This is where we're using chemicals to control our growth of microorganisms. We have 3 different ways. We have bacteriostatic, CIDAL, and lytic. So these curves with the growth and also the time out here, and then we're having actually 2 colors in each curve. We have the blue, which is the living cells, and then we have the red, which is the cells in general. So if we're taking this bacteriostatic, what do we look at? Yes, we have starting up with the growth, and here we are putting this bacteriostatic agent in the environment. Then the growth is inhibited, and when it's removed again, the growth can continue. So it's reversible in this bacteriostatic. If we're taking this bactericidal, then we have starting up with a good growth, then we're putting this agent into the environment. The living cells will die, so it kills the cells. The red one is still up here, and that's because the cells are still available in the environment, but the cells are dead. So this condition is that it kills, so there's no growth, but the cells are in the environment. This is irreversible. Then we have the bacteriolytic, where we again have a good growth. Then we are adding the bacteriolytic agent here, and the cells are killed. So this bacteriolytic agent is killing the cells. The red one here is also going down, and that's because of that the cells are lysed. So it's also this lytic is that the cells are destroyed, so they are not available any longer in the environment. This is also a irreversible agent. So this is where we can use chemicals to control our growth.